Today, I will present the application of CRISPR-Cas9 in plants. My name is Xing-Liang Ma. Thanks very much, LabRoots, give me the opportunity to present my work. First, CRISPR is a very popular tool for gene editing already. Now I will talk about the mechanisms of CRISPR, the Cas9 protein, and the sgRNA as a dual complex can create a double-strand breaks in the chromosome, which would induce mutation through the DNA repair system in the cells. It's easy to show that CRISPR is very popular, but still you need some vector to deliver the CRISPR system into your interested organism. Here I will present my CRISPR vector system that should be interesting for plant research community. And secondly, I will present my DSD method for decoding CRISPR-Cas9 induced mutants. Sorry, I will explain what is DSD stand for in my following slides. The first common component of CRISPR is sgRNA. So we designed the sgRNA intermediate vector that includes the sgRNA promoter and sgRNA backbone and Pauline T terminator. Since it's very small, so it's in a PUC 19-based vector. We cloned 4 sgRNA promoter for monocots: OSU3, OSU6A, OSU6B, and OSU6C. You can use these promoters for monocots plants. Then we cloned 4 sgRNA promoter for the dicots, like ATU3B, ATU3D, ATU61, and ATU629. Those promoters are better to use for dicot plants. In order to integrate your target of interest to the intermediate vectors, you can synthesize the adapter, the ligate to your to our intermediate vector, or you can use overlapping PCR. Here I need to mention, because the cassette overall is very small, generally it's around 500 base pair to 1 kilobase pair, so it's very small. So here we are using PCR to amplify it instead of clone it firstly. In this way, you can assemble multiple sgRNA cassettes by PCR at the same time, which is very convenient. So next step is the Cas9 part. Cas9 is big. It's around 4,000 nucleotide coding sequence, but you do not need to change it. And the target information is located in the sgRNA vector part. So we are doing this work for you. So based on the SP nucleotide sequence and the amino sequence of Cas9, we code optimized it for plants and then designed 99 overlapping oligos to assemble the Cas9 coding sequence with the nuclear localization signal attached on both ends. Then we confirm the Cas9 our version of Cas9 expression and the localization within the protoplast system. Here you can see we attached the GFP coding sequence to the Cas9 cassette, which is driven by ubiquitin promoter. Then we transferred this construct into protoplast. You can see the GFP signal localized only in the nuclear area, where the chromosome also present so that they can get in touch with each other and do the gene editing work. After established the Cas9 cassette, we integrated in the binary vector system so that you do not need to any change. You do not need to do any change on it. Regarding the selection markers, this is always a matter that our people doing transformation need to take care of, because different plants might prefer different antibiotic selection method. Here we have the version of NPT2, HPT, and bar gene selection, so that basically that means it has 3 vectors, so that you can choose which one would you prefer. And for the Cas9 coding sequence, you can find the ubiquitin promoter version and the 35S promoter version. So in total, we have 6 binary vectors for you to choose from at Gene website. For the cloning the sgRNA cassettes, we have BSA1 set, 2 BSA1 sets integrated in the binary vector, and so that you can integrate 2 to 8 sgRNA cassettes simultaneously using the Golden Gate cloning method. If you are not familiar with the Golden Gate cloning, you can refer to my publication later. I will also include in the reference part. With this CRISPR vector system, so we transferred to the plants, and we got a lot of mutations. Then we can analyze the mutation character induced by Cas9. Seems half of the mutation it's a single base pair insertion, and the remaining are mostly small deletions. This is important because for us to for the second part of the DSD method for decoding the CRISPR-induced mutations, we'll utilize this trait. So here is an example of the mutations we will get from CRISPR. Actually here is the res sequence. The wild type. Here we designed the targets in the bold sequence, and we got 2 mutant alleles types within the diploid plants. When we want to amplify this target region, then we and send it for single sequencing, we got the chimeric chromatograms. Upstream of the mutant set, it's totally normal and clear peaks, but downstream of the mutation area is overlapped peaks. If you look clearly, they are the combination of the mutant allele 1 and the mutant allele 2. Yeah, this is interesting and obvious, but what if you only have the superimposed chromatograms and you cannot read it directly? So that's why we are inventing the DSD method. First, we summarize the category the mutant types into 5 types. Can have insertion, and another allele can be still be wild type or deletion, or a combination of a single nuclear insertion and deletion. But all of them start, if you send it for sequencing, single sequencing, they all have overlap peaks starting from the mutation set. Of course, you can clone the PCR product and then send it for sequencing with multiple clones, so that you of course then you can know exactly what the PCR amplicant mixture can be, but this can be costly and not efficient enough. So by then we are already thinking, can we decode this overlap chromatograms without sequencing, without further clone it and then send it for multiple clones for sequencing. Eventually, we invented this method called degenerate sequence decoding. What you do is once you get the chimeric superimposed chromatograms, you'll find the anchor set, which is upstream of the overlap peaks around 15 base pair, and then you manually read the overlap peaks, and then convert into degenerate sequence. For example, you can convert C and T into Y, C and A into M, C and T into Y again. And so next, you query the degenerate sequence to the reference sequence. Once you find hit, you join the hit with the anchor sequence. In this way, you can decode the allele 1, and then you subtract allele 1, the blue part, from the degenerate sequence. And ultimately, you decode the second allele. Of course, this method is used mainly for dicot plants, including rice. This method, we use firstly, we invent in the manually manner, but a lot of our users are not satisfying about this. I think it's still tedious and require a lot of energy if you have hundreds or thousands of sequence for decoding. So we decided to develop a software to address this problem. It is called DS-Decode-M, and which is developed by Dr. Xianrong Xie. We worked together. In addition to this tool, Dr. Xie also integrated the other bioinformatics tools that will be used when you are applying the CRISPR system in plants. For example, if you want to find which part you can use for designing CRISPR targets, you can use target design tool. And if you are not sure about designing the oligos or primers that will be used for the targets, you can use the primer design. If you're worried about off-target that potentially exist in other loci of your genome, you can use the off-target tool to make sure that your targets are specific enough. With the CRISPR vector system and the bioinformatics tool called CRISPR-GE, we applied these systems in rice, which is main focus from my previous lab in Guangzhou. Here, the rice, there is a glutinous rice. You can see it's milky, but the rice generally we can find from the market, they are semi-transparent. There are only one gene that's different from the normal rice and the glutinous rice. The ratio of two kind of starch, amylose and amylopectin. Amylose is the ratio. What the ratio is 20 to 30%. It's the normal rice, and once it's not existing or very low content, is glutinous rice. The gene responsible for amylose synthesis is Waxy gene, which is already widely characterized. So in order to create glutinous rice from the normal rice varieties in a fast and efficient manner. So we designed 2 targets and in the extron of the Waxy gene. And then we transformed rice. It's in the T0 generation, which is the first generation of the transgenes. We already got the glutinous, obtained the glutinous rice. That means the 2 alleles of the Waxy gene are both targeted. Then we measured the amylose content, and then we found that indeed the amylose content is comparable to available glutinous rice from the market. They are not distinguishable. The next interesting question, and also the powerful part of CRISPR, that the target you designed is your target, of course, but the transgene cassette that will be integrated in the genome is random. That means they can be segregated from each other. So they are. Here I will show you why we can segregate the transgene, the transgene that contains the Cas9 cassette from the mutation of your interest. Firstly, since it's random, that means it's most likely that the tDNA and the target are not linked with each other. From the left in the illustration, you can see that still after 1 generation of Seifing, that 1/4 of the progenies will be transgenic free, where still they contain the mutation. But a lot of people are still asking, well, sometimes it can be possible that the CRISPR cassette will be integrated in the target set, and they will be linked with each other. And then how can you separate it? But we have to remember that the plants are generally diploid or polyploid. Even the Cas9 cassette are linked with one of the alleles. The other allele can still segregate without problem. Still, from the illustration on the right, you can see 1/4 of transgenes still, the progeny will still be transgene free. This is really the powerful part of gene editing. Here is an example of how we segregate the transgene fragment by Seifing. You can see 2 of the progeny lines, the 1 to 10, they still have transgene, and the 1 to 3 to 1 already lost the antibiotic resistance. That means the transgene part is gone. And but when we analyze the seeds from the 3 to 1 lines, you can see still glutinous rice. That means the mutation is still there, but the transgene part is already segregated. We spread this vector and construct to multiple laboratories, and they applied to other very interesting species. For example, tomato, rapeseed, kiwi fruit, cotton, poplar, etc. Here is some of the publications. Here is some publications resulted from our constructs. So the take-home message is that we developed a CRISPR vector system that is user-friendly and accessible through any gene, so that the general laboratories without any molecular experience can still use this vector system. And secondly, we developed the CRISPR-GE toolkit for you to design in the CRISPR experiment and analyzing the mutants from the gene edited plants. Here is the references from which I used during the presentation. If you are interested in more information regarding CRISPR, or I might missed something from my presentation that I didn't explain clearly, you can refer to my review paper, the CRISPR-Cas9 platform gene editing in plants, developments, and applications we published in Molecular Plant. Okay, I think that's all for my presentation. Thank you, everybody, for listening to my talk, and thanks LabRoots for giving me the opportunity to present here. Thank you.